Concept 25.5 says major changes in body form can result from changes in the sequences and regulation of developmental genes. So far we've been talking about some major events that are all external events. Things like mass extinctions and plate tectonics and adaptive radiation to some degree are all caused by kind of external events to the living creature. And the fossil record tells us the great changes that can be brought about by some of these events. But we can also understand how a lot of these major changes occur if we look internally, if we look at the intrinsic biological mechanisms that kind of control changes in living creatures. So what we want to look at is some internal genetic mechanisms that provide for change on a large scale evolutionary level. You would think that to see some of the major changes that we've seen throughout the history of the Earth, you would also have to have extremely major changes in the genetic makeup of creatures. But what we found as we've continued to study this is that's not always the case. Back in chapter 21, we talked about something called Evo-Devo, evolutionary development. It's basically research where the connection point, the interface between evolutionary biology and developmental biology, that is beginning to show us how very small, slight genetic divergences can produce major body morphological and anatomical differences between species. So you don't always have to have a tremendous change in genetics to get a very large change in what you see in the morphology of creatures. There are genes that program the developmental control, the rate and the timing, the arrangement of organisms' forms as it develops into an adult. So you have genetic information that controls, like for example, how many arms you have, and you have genetic information that controls where those arms are placed and how quickly those arms grow. Those are like control genes that control other genes. And minor changes in those control genes can lead to major changes of body shape and size and morphology. One such change is basically just in the rate and the timing. There's something called heterochrony. Heterochrony is the evolutionary change in the rate or timing of developmental events. Heterochrony is from the Greek hetero, meaning different, and chronos, meaning time. So this can have a pretty significant effect on the final outcome of the body shape. So for example, we can look at the skull of a chimpanzee and the skull of a human. The shape of an adult human and the shape of an adult chimpanzee skull are pretty different, but if we look at the shape of an infant baby and an infant chimpanzee or an embryo chimpanzee or an embryo human, we'll see that they're actually very much the same. So let's take a look at a picture. We'll come back to this. So here, if we compare an infant chimpanzee to a human fetus, a chimpanzee and a human fetus, you see the skulls are actually very similar. But what happens is, over time, there's a growth pattern that's quite different. In the chimpanzee, you have major growth of the jaw. And in the human adult, you have less growth of the jaw. So a human adult looks a lot more like a human fetus. A chimpanzee adult does not look much like a chimpanzee fetus. And you can see that in the picture. If you compare this mom and the baby here, the baby looks much more human-like. Its head is much more human shape than the adult chimpanzee is. These differences we see are based on the timing and the rate of change. So here we have a very low rate of growth in the jaw area, where here we have a much faster and higher rate of growth, and that ends up making these big differences that we see here. Those major differences, the contrasting shapes of the human and the chimpanzee adult skulls, are the result of really small changes in the relative growth rates. It's called allometric growth. So a small change in the growth rate eventually over time, over 18 or 20 years, builds up to some major differences in the way that it looks. If you want to watch a quick video, you can right click and hit play. This is about that allometric growth, which is basically differences in the rate of growth or development for different body parts or between different species. Another example that we can see with this heterochrony is how it can alter the timing of reproductive development relative to the development of the non-reproductive organs. What we mean is that in some creatures it may be an adult in all of its reproductive capacities. In other words, it has all of its various parts, like in a human we might consider post-puberty. It's able to reproduce, but it might still have certain characteristics that are very similar to an infant. So for example, there is a salamander that we're going to look at, and I'll show you a picture that has this occur. In the salamander, we have what we call paedomorphosis. Paedomorphosis is the rate of reproductive development accelerated compared to somatic development. In other words, the creature becomes sexually mature but still retains some of the body features of a child or infant variety of that particular creature. And the example we're going to look at is this salamander that's on the next slide. It is an adult, sexually speaking. It's sexually mature, it can reproduce, but it still retains like the gills of what you would consider a juvenile. Usually in salamanders, the babies, the juveniles, have gills, but as they become adults they lose those gills. Well, this one has a different rate of growth. Its reproductive system is fully mature, but it still keeps some of the juvenile features of its physical form. So that's another good example of this heterochrony. In this case it's called paedomorphosis, where the reproductive development is faster than the other body parts, basically. Not only is timing and the rate of growth an important factor, but another important factor is spatial pattern. In other words, big evolutionary changes can result from small changes in genes that control the placement and organization of body parts. Imagine if there were just a few genes that controlled where your arms are placed or the number of arms that you have. Well, a minor change to just those few genes could cause some major appearance changes to you. These are homeotic genes. Homeotic genes determine features such as where wings and legs are going to be placed and how they're going to develop on a bird, or how a flower's parts are arranged. So these are like control genes, and they control a large portion of what you see. So a change in one of these control genes could lead to a huge variety of changes in the anatomical features of the creature. A good example of these homeotic genes are called Hox genes. Hox genes are a class of homeotic genes that provide the information that tells the creature that is developing as a fetus where to put certain body parts. If Hox genes are expressed in the wrong location, you end up with body parts in the wrong location. For example, in crustaceans like a lobster or shrimp, a swimming appendage can be made instead of a feeding appendage. So you might have what's supposed to be a mouth part, but it comes out as a swimming part, a leg or a flipper part, and that's because of a minor error perhaps in these Hox genes that control the placement of the body parts. Here's a couple of amazing pictures of a snake embryo and a chicken embryo. The snake embryo on the left and the chicken embryo on the right. The purple that you see is the Hox gene expression of a Hox gene called HOXC6, and that correlates with a limbless region in the torso of the snake and the chicken. So you see in the snake it has a limbless region basically throughout, so it's not going to have any legs throughout here. And in the chicken it has a limbless region right here, so you'll have limb placement above and below that point. So there are certain genes that are like master control switches that tell the developing embryo, okay, this is where you're going to put the arm, or this is where you're going to put the leg, or this is how many ears you're going to have. And a minor change in that gene can cause obviously major differences. One good question that scientists have had for a while was this genetic diversity that was present to allow for a variety of these changes was already present before the Cambrian explosion. So the question is, the tremendous increase in diversity that happened during the Cambrian explosion, why did it happen at that point? Why did it not happen prior to that if the potential genes for diversity were already present? One potential answer to that may be these Hox genes, these developmental, these homeotic genes that we're talking about. Just like you can have various types of changes or mutations in other genes, you could have changes or mutations in these master control genes. And a mutation or a change in this master control gene could then cause a major kind of a cascading effect that might lead to very important changes in the creature itself. And then that could lead to potentially new physical traits or new adaptations that then could be acted on by natural selection, and then you might have rapid adaptive radiation and the development of these new novel traits. Say for example, having a particular flipper shape or something like that. It could be the result of a minor mutation in one of these control genes, and then that leads to a change in the flipper placement or the number of flippers, and suddenly you have a creature that has this advantage, and then this advantage can be acted on by natural selection and passed on. So perhaps that's what happened in the Cambrian explosion, was perhaps you had a change in some of these control genes that suddenly allowed for a wider variety of diversity and then a larger variety of anatomical features that were acted on. A good example of how this might occur is in insects. Likely new morphological forms come from perhaps gene duplication events that produce new developmental genes. So gene duplication events are not that uncommon. It happens. But what if it happens to a developmental gene, say a gene that turns off leg production? So now suddenly the creature has multiple copies of this turn off leg production gene. Well, that's a possible way that they've actually proven in the laboratory for the evolution of six-legged insects, because six-legged insects originally came from crustacean ancestors that had many more than 6 legs. Well, how did they go from having many more than 6 legs to the simple 6 that they have now? In the laboratory, they've shown they actually tracked it down to a specific nucleotide sequence and a specific nucleotide change in a particular gene called the UBX gene. It's a developmental gene. It has been identified that can turn off leg development. So a very small change in the nucleotide sequence of this one developmental gene basically has affected the outcome of what you see, going from many legs to perhaps just the 6 that we see in the insects. So if we're talking about these control genes, these Hox type genes, a small change in a nucleotide sequence can lead to large morphological results. Your textbook shows a diagram of how this could have happened. About 400 million years ago, you had a particular change in this Hox gene 7, and that led to a developmental change where basically the number of legs was suppressed. And so instead of having many legs like the crustacean ancestors of the insects, you have only the 6 legs that insects now have today. Alright, let's take one last break here and then we will finish up. We only have one small little concept left, and I'll save that for one last one just to make this one not so long. So we'll stop right here. We'll come back for the final concept for Chapter 25.